Committee of the Taxonomy of the Viruses. She is one of the members in the Geminiviruses, and today she is going to speak an insight into the resistance to Begomovirus. So I would like to now invite to Dr. V.G.Malathi. And Dr. Malathi, please, now floor is yours.
Madam, unmute it.
Unmute yourself. Unmute.
Thank you very much, Dr. Dubey, for all those kind words. And good evening, everybody. Can you stop sharing your screen, and then only I can put mine.
I thought you were sir.
Is it okay?
You have to slow the presentation.
Yeah, I have gone to presentation mode.
No, it is Zoom.
Is it Zoom?
Sir.
First open your slide on the desktop and then you have to share.
No, you have to first stop your sharing, no?
Madam, this is your sharing. Already V.G.Malathi is sharing.
Madam,
one minute, I'm doing it.
Yeah.
Is it okay now?
Yes.
Yeah.
Yes, madam.
Fine.
Yes, madam.
Yeah, that's good.
Okay, thank you all very much. See, my talk is going to be away from the cassava and the tuber crops. And there are two reasons, and I should thank Sheila and Makesh for it. Number one is that in CTCRA, we do gap filling whenever one stem cutting doesn't germinate. So initially, I was called more as a gap filling because Maruti had some problem. But then later, I think Sheila and Makesh felt that it's okay, we will call her. So I'm here, and I think I'm more here because of the love that I have for cassava mosaic viruses. Okay. It's not going next. 1 minute. Okay. This is just to recall that those days were the serology days when I worked with the antibody of T strain and the K strain of ACMV and found that Indian cassava mosaic virus, when it reacts, it has got a spur. And this was the first picture of Geminivirus for India from ICMV infected variety Kalikalan. And of course, I'm always thankful to John Stanley for introducing me to this world of Geminiviruses. Okay, so today, I know that many of you are in cassava, but at the same time, you are also working on other crops. So what is the difficulty? Like Stefan really put it very clearly, the resistance breeding part of it for brown streak virus, how it is difficult. So I would like to share with you some of the latest information and how badly we are prepared in achieving resistance for Begomoviruses. I'll briefly outline. So let me describe the Begomoviruses. There are certain conventional dominant R gene, but most of them are recessive resistant genes. But increasingly, metabolic genes are being assigned to this work of resistance. And the most important, as Stefan and others put it, is that the screening for resistance. What is the protocol that you will be following it? And that is really difficult, to pinpoint what is the kind of inoculation you will be doing it. So this we are all familiar with, the kind of diseases, yellow mosaic, leaf curl, and all those diseases, the whitefly and the geminate particles. So this is to tell you all this today, because it is cassava, there was no mention of the satellite elements, that it has got the distinct DNA, A and B, that is a story. But when you come to the Indian subcontinent, especially the old world, other Asia, China, and other continents, and in other crops, we are having 3 types of satellites which encapsulated independently in an independent molecule. That is, the code protein is from the Geminivirus only. but it is not a geminate particle, but just an icosahedral incomplete particle in which this alpha satellite, another satellite is beta satellite, and another half of this molecule is delta satellites. So there are three types of satellites associated with either DNA A alone or associated with both DNA A and B. And these are symptom modifying molecules. As a result, it complicates any disease scenario, and definitely it has complicated the resistance screening also. And before I get into the resistance screening, normally what we do is we have to understand the viral pathogenesis thoroughly. But surprisingly, in spite of the last 30 years of work on Gemini viruses, there are more gray areas rather than a clear-cut black and white areas. For example, for any pathogens, we are now very clear. There are pattern recognizing receptors. That is, the pathogen SPAMP molecule will be recognized by the receptor molecule, and that very recognition will be given signal, resulting in the cascade of defense metabolism. And this is a very broad spectrum resistance present against all pathogens in most of the plants. You can even call this as a basic immunity or a even non-host resistance. But we do not know anything about whether there are any PRRs or pattern recognizing receptors. Nearest to this point is a recently discovered gene in tomato against tomato yellow leaf curl virus, which is called as the barley anytime gene. And this is considered to be a PRR, but it is yet to be proved. Other than that, there is nothing known about it. So virus enters mostly because the vector is directly putting it inside the phloem parenchyma and cambium cells. But it has to disassemble, release the coat protein molecules and release a single stranded DNA. How it is facilitated? Like, for example, between resistant and genotype and the susceptible genotype, do we have difference either in the cell content or in any other particular gene is involved that brings about the disassembly of the particle? Generally, in virology, we take it that it is the pH of the cell which decides this disassembly part of it. But at least we should know what is differentiating a susceptible genotype from the resistant genotype, and this is again a green area. And the ssDNA, single-stranded DNA, has to enter the nucleus. It cannot do anything in the cytoplasm. So how does it enter? Like, we take it that any cell cycle protein like karyofurin or importin or any such protein, cytoskeleton protein, must be mediating the entry of the DNA into the nucleus. But again, we are not sure about it. And this single-stranded DNA should get immediately packaged with histones and become a mini chromosome and become a double-stranded DNA. That is, the single-stranded DNA should be copied into another strand. Who does it? It is only host DNA polymerase which does it. But do we have any information on host DNA polymerase profile between the non-host and the host, or between the resistant and susceptible genetic? Is it possible that irrespective of whatever the genotype, the viral first replication cycle will be initiated? This is again a gray area. And then what happens is, the double-stranded DNA is the template, is the replication competent and the transcription competent one, and which will be producing the mRNA and the translation products. And then comes the movement. And when it comes to movement, I just do not understand. We do East2 hybrid system to pick up the host proteins. But I find that many host phloem specific proteins are still to be characterized, which will have to react with Gemini viral movement protein. Let it be the monopartite movement protein or the bipartite movement protein. And then once it reaches, whether the virus moves in apoplast or in symplast, are there any transporters protein involved? Like, increasingly now, even the large molecules, proteins, etc., to cross the membrane and to go between apoplast and implast, there are transporters, like sweet transporter is the one which plays a major role in rice against the rice pathogens. So such information are still lacking for Gemini viruses. And unless all this molecular mechanism of how the Gemini virus pathogenesis is executed are very well understood, it is going to address the issue of resistance. And of course, the host factors, whether they are involved in any transmission or not, is not yet known. So this is only to give a brief introduction. I'm not going to take much time. So this is the core protein and this is the replication protein. But the very important pathogenicity determinants are the AV2 protein, AC4 protein, and AC2 protein. Mainly because they suppress the RNA silencing, and so they have gained a lot of importance. But what I would suggest, RNA silencing alone is not the defense system of the plant. So for all the satellites, in the case of beta satellite is a very important one, because in the presence of beta satellite only, we get very severe symptoms. So the only protein that is encoded by the beta satellite, the beta C1, is another pathogenicity determinant. And so how the virus is facing these determinants are there to cause the disruption in the host. And how the host is facing these proteins will constitute what we call as resistance. So people have started looking for the resistance. Of course, the typical way is to do the mapping population and go by markers and then arrive at the gene locus and then the characterize the gene locus. But increasingly, what we gain nowadays is what we call as reverse genetics, where you pick up from the protein. So one way is you can try to find out which are the host proteins reacting with the viral protein. And we can go for the host proteins which are exclusively expressed in phloem. And many important cellular processes, like for example, the DNA repairing, DNA synthesis, or even cell division, all those very important cellular, any energy releasing mechanisms or pathogen or metabolism, you may have to look into. So all these approaches have been taken into consideration, and some of the genes have been really characterized. So we can sort of group the genes into those which are actually involved in RNA silencing, and those which are involved in what we call as a recognition-based R protein. That is, we are all very well aware of it in other pathogens, but in virus pathogens, very few cases only are there where the R protein recognition-based resistance mechanism is suggested. And increasingly, the autophagy is one where the host degrades the system of proteins, which will not be necessary at all for the development. So whether the autophagy is involved in such viral resistance. And now, in very recently, translational repression seems to be a very important mechanism by which the virus can be resisted. And of course, the most important defense trigger or the cascade trigger is a MAP kinase interactions. And so MAP kinase is another one. Other than that, some metabolic genes also have been assigned some roles. We'll quickly go into what of these. For all the literature on Gemini viruses, you will be surprised to know that so far, only 2 R genes are known. When I say R gene, the conventional nucleotide binding sites are leucine-rich repeats with the N-terminal region, either coil-to-coil amino acid or tall interleukin-like receptors. Only 2 R genes are known, and that I thought is very surprising. Either we must be going in a very, very diverse way. The first R gene which was characterized was against Mungbean yellow mosaic India virus from India, from Calcutta-based institute, and they got it in Mungbean, and they got this R gene factor by doing lot of molecular markers they had, and then they shortlisted it, and then the locus was completely genome sequenced, and then they found out this. And it has got a typical coil-to-coil domain and a leucine-rich domain. And interestingly, they did certain computer simography, and they found that it's a horseshoe-shaped N-terminus and the C-terminus domain, and into which they found out that the CP nicely fits in. It is by molecular dynamic study. So this is the first R gene which was characterized. But unfortunately, they didn't take it further, means they have not knocked it down, or they have not shown by transgenic that the resistance is being conferred and all that. But they suggested that such a gene is present. But the most interesting work is the TY2 gene conferring resistance against the Begomovirus, tomato yellow leaf curl virus. Those who are working in tomato or any solanaceous crop, we are all very familiar with a bunch of genes, TY1, TY2, TY3, TY4, TY5, TY6. And for a long time, this TY2 gene was not characterized. And interestingly, this TY2 gene is a contribution by our earlier Deputy Director General Kallu in Habrochitis tomato. He only got this variety having this particular gene. And this TY2 gene was not at all characterized for a long time, but last year it has been characterized. And this has also been found out to be a typical NBS type with the leucine-rich repeats. And so they compared the sequences and they found out that the one that is having the R gene conferring resistance MYMV is totally distinct and different from the one that is present in Habrochitis and tomato, which is conferring resistance to tomato yellow leaf curl. But one step further they went, they made a transgenic plant, the highly susceptible transgenic plant, they made it, and then they challenged inoculated by agroinoculation, the TYLCV, and proved that, yes, this is the TYT gene, and this is the one that is giving resistance. In fact, this is an absolute proof given, to say that this gene is involved in resistance. So this is just to summarize this, like we have TY1 and TY3, which are actually RNA-dependent RNA polymerases, which are involved in the RNAi mechanism. And TY2, now I told you that. And TY4 is not yet classified, but TY5 is a very, very interesting gene. It is a recessive gene, but it is one of the mRNA surveillance factor. For example, in any system, when ribosome is synthesizing the protein, the ribosomes always get recycled, and there is always a surveillance going on. But they found out that this recessive gene, means by mutation, it would have become a recessive, and this gene is not able to recycle the ribosome, and that is why the viral mRNA transcripts are reduced, and that is the kind of the resistance it can give against TYLCV. And here also they achieved everything, like doing agroinoculation, transgenic and inoculation, and further proving that the resistance is held on. And the TY6 is again not yet characterized. This is the most important kinase gene. In fact, the cell energy metabolism, the level of ATP, everything is maintained because of this kinases. Because in any cell, the reactions are held up, homeostasis is achieved mainly due to phosphorylation and dephosphorylation. People who are familiar with the signal cascade, you know that it is only the switchover from the dephosphorylation to phosphorylation that the molecule gets either inactivated or activated. It is passing of the phosphate molecule or kinasing of the phosphate molecules. So in that respect, it was shown that the RK1 has got a kind of conferring resistance both in TYLCV and Cabbage leaf curl virus. Then this is one of the permease-like gene, and the transporters, that just as I told you, transporters are going to play a major role in understanding the viral pathogenesis, because these transporters only facilitate the movement of the viral protein and also viral genome across the membrane. So definitely they will have to interact with the transporters. And this is another protein, which is lipocalin proteins, which are involved in the resistance. So this is a transcription factor. In fact, sometime back, many of the transcription factors were given the resistance role even against many pathogens. But when the transgenes are made, it is not, foolproof, and then the concepts are not very clear how this transcription factor alone can bring about the resistance. The interesting case is the nuclear shuttle protein interacting kinase, which we will be dealing with it very elaborately in the forthcoming session. This I'm not touching because many people who talked on cassava, they had a very good sum up of these two genes. So coming to T/1 and T/3 genes, and once it has been sequenced and characterized, and Verlan et al. Showed that it is an RNA-dependent RNA polymerase of the gene that is enhancing the production of siRNA. By using various siRNA as the primers, it is producing lot of double-stranded RNA, which is generating to more number of siRNA. The more number of siRNA when it is produced, the RNA-dependent methylation system is getting switched on. So they told that the mechanism by which this T/1 and T/3 perhaps bring about the resistance is the viral genome is totally getting methylated. Methylation takes place, naturally the replication or even any of this transcription and translation and all will be inhibited. As a result, the viral titer goes down. So this is the case of T/1 and T/3. I am glad to tell you that I was associated to some extent using these gene strategies for the resistance. And this work was done with Dr. Prasanna from AVR. Now he is in IHR. What he did was he pyramided the genes T/1 and T/1, T/2, T/2, and T/1 and T/3. And what was the opinion held till then was that T/1 and T/3 are able to hold to some extent the bipartite virus, which is tomato leaf nudity virus. Whereas the resistance which was due to the T/2 gene could not hold against the bipartite virus. So this, to prove this and then to arrive at the pyramiding of the genes, what he did was he used all our clones to inoculate and do the glasshouse inoculation and then come to the field for screening. And this is the thing, the pyramidal lines with the T/1, T/2, T/3 pyramidal lines. And these are the susceptible variety, which showed that definitely the pyramiding of the lines will give you the resistance. The other group of resistant genes is the autophagy related genes, because you know that the autophagy is only nothing but the degradation of the pathogen protein so that further the pathogen cannot be established. So initially the proof was given that the autophagy ATG5 and ATG7, when you are making applying it by auto agrobacterial regulation, the resistance was conferred. Whereas the gene cluster GAPC, when it is transiently expressed, it increased their resistance to viral infection. So they said that the antiviral mechanism can be achieved by incorporating any of this ATG related genes. The other gene which is very, very important is the gene that is nuclear shuttle protein interacting kinase, which was initially isolated from Arabidopsis. Now, you know, the homologs of it are getting isolated from many plants. What happens in a normal plant is this NIK, again phosphorylation. After it gets phosphorylated, it will phosphorylate another ribosomal protein. And this ribosomal protein will enter into nucleus and will bind to the MIB transcription factor. As a result, the translation will be repressed. So the translation repression is emerging as one of the very important mechanisms to achieve the resistance. And this I have graphically represented. Like for example, NAKV1, when it is there, then this NSP is also there, and this NAKV phosphorylates this RPL10, and RPL10 will go and attack the MIB translation factor so that, you know, the repression of the gene takes place. So the translational repression was first time shown working against, say, Gemini virus resistance. And what they did further, in fact, this was done by Rodenstein from North Carolina University. What they did was they made again the transgenic with the NIK1, that is the nuclear shuttle protein interacting kinases. And once they made the transgenic, and then the inoculations were done with the cabbage leaf curl virus, and they showed that definitely there is a resistance. But the problem is the penalty, because you are repressing the translation. So naturally the fruit size and everything was small. And then, the growth of the plant also was very highly reduced, but definitely it was conferring resistance. So the other major enzyme is the MAP kinases, because we know that in any signal transcription triggering, the initial MAP kinase 1, MAP kinase 2, MAP kinase 3, and they get phosphorylates and dephosphorylates, and then finally they phosphorylate the WRKV transcription factor, which will enter into the cells and will increase the transcription of the other genes, the defense genes. So the jasminic and salicylic acid related genes like PR1 and SAPN1 and SAPN2, all these were enhanced whenever the MAP kinase activity is increased. Naturally, that is because there is an increased peroxide accumulation, superoxide dismutase action, peroxidase action, all these which are giving rise to the superspecies of reactive oxygen, and thereby, bringing about the highly energized metabolism. So overall, the research suggests that perhaps MAPK3, that is nitrogen activated kinases, also could function as a resistance factor. In fact, they again made the transgenic plants and showed the resistance. This is the wild type. And then, they have got several events in which they showed the transgenic resistance, and the relative level of the virus load was very low. And that is how they proved it. To sum up, until today, what are all the defense genes available to us from the host? Say, for example, on one route is the autophagy route, where, the beta-7 protein is known to react with ATG8. So if the host will always see to it that the autophagy is switched on, but the beta-7, and that is why it's binding to it and cuts it out. So one route possible is autophagy. The another one, as I told you, the phosphorylating systems, which phosphorylates different proteins. And these proteins are getting degraded finally by proteasome reaction, 26X proteasome reaction. So there are many Gemini virus related interacting kinases known, which play a lot of role, to inhibit or to promote the kinasing and also to prevent the proteasomal interaction. The most interesting one, as I told you, is the NIK1, which will be doing the phosphorylation and thereby the BIP translation factor and then the translational suppression. So the polyribosome binding of the messenger RNA will not take place. But besides that, there is another protein, again, it is interacting with the nuclear shuttle protein, but it is one of the GTPase, which is responsible for the export of the viral nucleic acid from the nucleus. What is happening is there is one protein called WW protein. W is nothing but tryptophan. So tryptophan proteins will bind to this NIJ and then will direct it to the bodies in the nucleus. As a result, it is not available for the export from the nucleus. So this is another method of resistance. Finally, it was shown by our Indian group, Manoj Gupta, where he showed that the protein RP24 binds with the intergenic region of the Gemini viruses and does not allow the RNA polymerase 2 to do the transcription. So it is kind of a multi-pronged attack the host is planning, one on autophagy system, another on proteasome system, another on movement of viral genome, the translational repression and transcription repression. So in all these ways, the host is having a multi-pronged attack to prevent the virus infection. So this is all regarding the genes. But being a virologist and often associated with the resistance screening, often there are problems which I thought I'll bring to your notice. Normally, what is done is that, either we field screen it in the hotspots, or we try to do the glasso screening, which Stefan said, even for a highly transmissible virus, to do whitefly transmission is very, very difficult. And so everybody resorted to agroinoculation, where the cloned constructs are mediated through agrobacterium. But there are certain hosts in which you don't get consistent success. For example, in cassava, and that is a photo myself and John, we were trying to achieve it. Like I tried to inoculate the ICMV, ACMV into cassava, and I never succeeded. Even now, I don't think the consistent inoculation protocol for cassava is available. So this is the agrobacterium mediated delivery, which is very much being used, at least in the case of cucurbits and tomatoes and even chilies to a great extent. But what I suggest is that the problem is like always these satellites are forgotten. And then, the inoculation is done only with the main virus. For example, tomato leaf curl, Bangalore virus. So the challenge number one is whenever we are doing field screening, do you know which is the virus against which you are screening? So for that matter, we should know which is the predominant virus. Maybe in the case of African continents, we are very sure, and now perhaps in India, for cassava, you are very sure. But for other hosts, I just do not have any idea what is the virus we are screening when you are doing field inoculation or field screening. The second is that satellites. Say, for example, TYLC BAB resistant lines were released by both IHR and UAC Bangalore having TY2 genes like Nandini and all those varieties. Once we had the satellite DNA and we inoculated along with the satellite DNA, I did not get any line which was resistant. So it is possible that when they did whitefly transmission, somehow beta satellite was not transmitted. As a result, the symptoms were very mild or they escaped symptoms. So there must be some problem in the consistent transmitting of beta satellites, which needs to be worked out. And as I said, the symptom-free plant. Dr. Imran also presented about the asymptomatic plant. Now the increasing trend is, the plants appear asymptomatic, but all of them have the virus presence. At least about 40 to 60% of the plants in the field, they are going to have the virus present. So is it okay if we just check it up by the symptom basis, or whether we have to go by PCR and ultimately again, re-inoculation and all those doubts are coming to in your mind. And the problem in the case of beta satellites is that it's not only for tomato viruses. I took example of tomato, but there are many more viruses there. One virus is associated with several beta satellites. And that is true also of Bindi yellow vein mosaic virus and Okra Initial leaf curl virus. So they may be associated with, in Karnataka, with the tomato leaf curl Bangalore beta satellite. But when it comes to Maharashtra, the Thailand beta satellites are there. And symptoms are very severe when it is associated with the Maharashtra with Thailand beta satellite. And the worst situation with the tomato leaf curl New Delhi virus, which at least in the field is associated with 4 different beta satellites. And with every beta satellites, it will be producing symptoms. So just imagine a genotype which may be able to withstand, one virus, one beta satellite combination. But with another virus, another beta satellite combination, maybe it will succumb to the disease. So whether do we have to consider all those variations also? And of course, in the we do know that the climatic parameters are very important, and many things we know about it. But what now I feel is that depending upon the climatic parameters, even the host gene expression, say for example, the resistant gene expression or any metabolic gene expression, that may undergo certain fine tuning or variation, which may reflect upon the interaction between the host gene and the viral gene, resulting in a kind of a range of variations in symptom prediction. And so definitely it affects the whitefly population. And the replication of the virus is very much affected due to the climatic condition. Even suppressor activity is very well known, like cassava Gemini viruses have got different temperature requirement to bring about the activity. And another things that are happening, the two Gemini viruses or Begomoviruses present together. If they are present together, one thing, their suppressors are actually mutually helping each other. And that is number one. And another one is that heteroencapsulation. And for example, like tomato leafle New Delhi virus and tomato leafle Palampur virus, they occur together in North Indian conditions. And we could easily show that when you are using TOLC-PA, it can encapsulate the TOLC-B and thereby producing a very good symptoms. So the heterocapsidation or complementation between the two Begomoviruses again is a common challenge. And the worst of all is the mixed infection with RNA viruses. In fact, it was very well shown that the TY1, TY3 conferred gene, the resistance was highly compromised with the CMV infection is present. And in fact, it is true about India and I'm true about many countries also. Like it is very difficult, like exclusively one virus alone present is very difficult. And always in cooker bits, you will have the WMB always with Begomovirus. So the mixed infection really create a problem compromising the resistance conferred by the known resistant genes. So to sum up, I would say, like, yes, the problem is the immense diversity of Begomoviruses, but our knowledge is highly limited, except discovery of these two R genes, because I'm sure that if we are capable of discovering R genes conferring resistance, to some extent, though I know it's a monogenic vertical, and a lot of people will say strainal variation, etc., etc. But I tell you, the monogenic resistance are the really good ones, which confer resistance at least for about 5 to 10 years of duration. So in the absence of those translation repression, as we have seen in the case of NAK-mediated resistance, and the metabolic genes for the kinases, which are involved in various phosphorylating and dephosphorylating genes, they could really play a big role in, conferring resistance. So way forward is first, we have to, that is because, we do not know how to screen it. Like, for example, this question we always debate in our project meetings. Like, is it necessary that we isolate the gene and maintain a separate white type and, screen only for one virus? Or the field screening now we do is okay. But somewhere, we have to have a compromise, and we should also look for a very critical way of working so that they are sorted out. And then the protocol, as I said, which virus, the quantitative measurement, and that is very, very essential to tell against which virus this particular gene is conferring resistance because of this mechanism. That we will be able to tell only when we have got a very precise way of measuring the resistance and the virus concentration. And of course, the population structure of the virus. So for example, morning we had an interesting talk, like one viral population displaced the another viral population. So that must be happening. Like we think that it is ACMV or ICMV or Sri Lankan Cassava-mosaic virus, but now suddenly, the Kerala, everything that you take sample is Sri Lankan Cassava-mosaic virus. And we should know what is the time lapse and how it has come to this situation. And how did it happen? Is it because of introduction of our varieties or any cultivation practices or climatic factors or the type of whitewave population that existed earlier and now it has changed? That we have to know. And then, of course, the elucidation of host factors which are facilitating the virus function and also interfering with the viral functions. Because if you know both of them, we can definitely achieve resistance. With this, I thank you all for your patient hearing. As I told you, mine was a little bit diversion, but more because of kindness of Sheila and Makesh. Thank you very much.
Thank you, Dr. Malathi. Very wonderful and informative.